Welcome to our exploration of CRISPR and genome editing. In this session, we will see how a natural bacterial defense system became a precise tool for rewriting DNA, and why that shift is reshaping medicine, agriculture, and biotechnology. Think of this as a guided tour from basic concepts to real-world impact, with a clear look at both promise and responsibility. Here is the roadmap for our journey. We will start with what CRISPR is and how it works, then move through key techniques and delivery methods. We will examine medical, agricultural, and industrial applications, pause for ethical and legal questions, and finish with current challenges, future directions, and a concise wrap-up you can take away. To begin, let's orient ourselves around CRISPR. At its core, it is a programmable way to find a specific DNA sequence and make a change exactly where intended. You can imagine it as a hyper-accurate search and edit function for the genome, opening possibilities from correcting single mutations to reprogramming cellular behavior. Genome editing means making targeted, intentional changes to DNA. Historically, we moved from selective breeding to early molecular tools and then to precision methods. As the timeline graphic suggests, the arrival of CRISPR accelerated everything by combining accuracy, efficiency, and affordability, turning difficult genetic modifications into routine laboratory steps, and enabling broader access to advanced biology. CRISPR's story starts in bacteria, where repeating DNA sequences and associated genes form an immune memory against viruses. Scientists discovered these repeats and connected them to defense, realizing they could be repurposed for editing. Pivotal milestones, including the demonstration of RNA-guided cutting, transformed a microbial strategy into a universal genome engineering platform. With that foundation set, let's dive into how CRISPR-Cas9 actually operates. This section explains the moving parts, how they find their target, and how the cell's own repair machinery completes the edit. Understanding the choreography behind recognition, cutting, and repair will make the later applications and safety considerations much clearer. The CRISPR toolkit has three key elements. Cas9 is the molecular scissor that performs the cut. A guide RNA provides the address, bringing Cas9 to the matching DNA sequence with remarkable specificity. And a short DNA signature near the target, called a PAM, acts like a landing pad, ensuring the system binds and cleaves at the correct spot. Editing unfolds in three steps. First, Cas9 and its guide RNA scan DNA until the PAM and matching sequence align, locking onto the target. Next, Cas9 makes a precise break in the DNA. Finally, the cell repairs that break either quickly, introducing small changes, or with a template, allowing accurate gene correction or insertion as the timeline depicts. Now that the basics are clear, we can explore variations on the CRISPR theme. Researchers have adapted the system to cut differently, to make edits without cutting both strands, and even to write new DNA sequences with remarkable control. This section shows how those innovations expand the toolbox and tailor it to specific scientific and therapeutic goals. Different CRISPR systems offer distinct strengths. Cas9 typically makes blunt cuts, while Cas1-2A creates staggered ends and recognizes different DNA signatures. Base editors chemically change a single letter without fully cutting the DNA, boosting precision. Prime editing goes further, rewriting short sequences with minimal damage. Together, these options let scientists match the method to the desired edit. Getting CRISPR to the right cells is half the battle. Viral vectors deliver efficiently, but must be managed for safety and immune reactions. Non-viral methods like electroporation and lipid particles avoid viruses and can be gentler to control. In vivo delivery edits inside the body, while ex vivo edits cells outside and returns them, offering additional safety checks. With the tools and delivery in mind, let's turn to where CRISPR makes a difference. We will look at breakthroughs in healthcare, transformations in farming and manufacturing, and ways the technology accelerates research. As the visuals suggest, these applications move from patient care to fields and factories, tying molecular precision to everyday outcomes. In medicine, CRISPR aims to correct inherited mutations at their source, offering durable therapies. It refines cancer care by targeting tumour mutations for diagnosis and treatment design. And it helps control infections by spotting or disabling pathogens, informing antiviral strategies and vaccine development. Together, these advances push healthcare toward more precise, personalised and preventive approaches. Beyond clinics, CRISPR strengthens crops against disease and climate stress, raising yields with fewer inputs. It improves livestock traits and resilience, supporting healthier herds. In biomanufacturing, edited microbes become efficient factories for fuels, pharmaceuticals, and specialty chemicals. The panels illustrate a shared theme: precision edits driving sustainable productivity across the bioeconomy. Powerful tools demand careful oversight. In this section, we step back to weigh ethics, law, and social impact. The goal is not only to ask what we can do, but what we should do under what rules and with whose consent. Responsible progress depends on clear norms and public trust alongside scientific innovation. Key ethical questions revolve around who is affected and how. Edits in somatic cells act on one person, while germline changes pass to future generations, raising higher stakes. Robust informed consent protects autonomy and guards human rights. Concerns about selecting traits remind us to prevent inequality and unintended consequences as capabilities advance. Regulation is still catching up. Rules differ across countries, balancing innovation against biosafety and ethics. Policymakers face uneven standards and evolving science, making governance complex. Public understanding varies widely, as shown in the panels, so education and transparent communication are essential to build trust and guide sensible, adaptable policies. Next, we focus on the hurdles that shape the path forward. Recognizing limitations helps set realistic expectations and directs research where it matters most. We will consider accuracy, delivery, and biology's own defences, then look at how emerging innovations aim to overcome these barriers and unlock safer, more effective gene editing. 3 technical challenges dominate today. Off-target edits must be minimised to ensure safety. Delivery needs to be efficient and cell-type specific, especially for hard-to-reach tissues. And immune responses to CRISPR components can blunt effectiveness or pose risks. Each issue is solvable, but together they demand better designs, smarter vectors, and careful clinical strategy. Innovation is advancing on several fronts. New enzymes and engineered guides improve target specificity and reduce unintended changes. Applications are expanding from therapy to agriculture and synthetic biology, where precision matters at scale. Integrating CRISPR with AI for design, with nanotechnology for delivery, and with synthetic circuits for control promises more accurate and reliable outcomes. We are ready to bring the story together. In the conclusion, we will distill the essentials of what CRISPR is, how it works, where it is used, and what must be managed. The aim is a clear, balanced picture you can carry forward, recognising both the extraordinary potential and the responsibilities it entails. In summary, CRISPR is a programmable, RNA-guided system that enables precise edits to DNA. Its mechanism revolves around targeted recognition, cutting, and repair, with variants like base and prime editing broadening control. Applications span healthcare to agriculture, tempered by ethical, legal, and safety considerations. Progress depends on sharpening precision, improving delivery, and governing use wisely. Looking ahead, the greatest impact may come from durable genetic cures and resilient, sustainable crops that reduce environmental pressures. Achieving this responsibly will require international standards, inclusive dialogue, and data sharing so benefits are equitably distributed. With collaboration and careful stewardship, genome editing can move from promising demonstrations to trusted, real-world solutions. Thank you for joining this deep dive into CRISPR and genome editing. I appreciate your attention and curiosity. If this sparked questions or ideas, that is the perfect next step, because thoughtful discussion is how we advance the science safely and effectively while ensuring the outcomes serve patients, communities, and our shared future.